Jeff, having worked on this project, together and over so many years, can you help put it in context for us as we've just gotten belzutifan, which is a HIF-2 targeting agent. It was FDA approved kind of end of last year, 2023, and starting to come out into clinic. So I think understanding the pathophysiology, what are, other Achilles heels, if you will, of HIF-2 signaling. So would you foresee, targeting of ACSS2, in combination or as a mechanism of resistance? Do you think that this is something that will ultimately, hopefully lead to drug development and patient care someday?
Yeah, I could see it. I can see it either in combination or in resistance. I mean, this is a different mechanism than belzutifan. So I would expect that they're going to work in concert to give a stronger response. So this might actually be a nice add-on to belzutifan. I mean, the other thing to add in terms of why ACSS2 is potentially intriguing target is, ultimately we think the mechanism of this is dependent on the enzymatic product of ACSS2, which is acetyl-CoA, and how it then modifies proteins to histones and chromatin for gene expression, but then other proteins like potentially HIF-2 itself for regulation of its stability. But there's multiple ways to make acetyl-CoA, but there's not a huge number. There's a limited number. There's 3, maybe 4, depending on how you add it. So different cells are going to have different dependencies on this mechanism versus others. So it's possible that kidney cancer cells, for whatever reason, are using this pathway, are more dependent on it. So there might be actually some sort of metabolic selectivity in terms of which cells are going to be affected. And Zach did a really nice job showing that if you knock this enzyme down or inhibit it, you actually do get quite a nice anti-tumor effect in at least some preclinical models and some in vitro models. So it does look like kidney cancer cells are going to be really requiring this one specific pathway. So it could be the kind of thing that, again, we'll have to see what kind of toxicity sort of profiles are out there, but there may be enough redundancy that other cells have plasticity and they would use other pathways, but it could be a selective vulnerability of kidney cancer cells due to their altered metabolism in the first place, that then gives you a second way to modulate HIF-2. And that could be an additive approach. And because the mechanism is different, I would expect that would be the case. But it could also be as in terms of resistance.
And I think you both mentioned it, and I want to highlight because I know you've both been involved in work understanding kidney cancer specifically in the tumor microenvironment as it relates to not just affecting the cancer cells, but looking at the immune cells. And so, Zach, you mentioned, you felt like this was really dependent in cancer cells. Have you looked at the immune cell populations in the RCC tumor samples or in your in vitro system to understand how it blocking ACSS2 would affect the immune cells?
We didn't share it in this publication. I do have some preliminary data that where it looks like treating with the same small molecule didn't have any negative effects. There were some potentially beneficial effects, but we would really need to dive a little bit deeper into that. There is some other literature that came out from Erica Pierce's group where they had looked at some acetate metabolism effects on, I think it was cytotoxic CD8 cells, where it appeared to be beneficial. The buildup of acetate, and they were kind of utilizing it a different way to become better at what they're doing. So I do think that there is definitely a selection or sensitivity to this particular metabolic pathway. Why that is a particularly interesting question that I think we don't really have a good answer to at this moment, but probably involves some other epigenetic regulation or something along those lines.
And Zach, where do you feel like this project might go next for you? Are you going to continue working on this or future experiments?
Yeah, I mean, I think it's certainly worth continuing to investigate and dig a little deeper. I think what would be really beneficial is to to take this system into a immune competent, syngeneic sort of mouse model to really kind of emulate what is happening in a patient. We could also dive into organoids and kind of see, from our patient pool, how do they respond? How do their different cell types respond? I think those sorts of analyses would be really important. And kind of taking this to the next step, we did look at 2D cultures of patient cancer cells that were, from patients we have here at Vanderbilt, and they did respond differently than the normal adjacent cells. So I think it would be really nice to kind of start parsing out how these different cells respond to, inhibiting acetate metabolism and really kind of fine tuning how this could be a therapeutic agent moving forward.
Well, that's exciting.
And
and maybe Jeff, I'll end with one last question with you. In, I think, cancer in general, we've certainly tried targeting various metabolic pathways. Maybe the best well-known story is IDO inhibition. And then I would say more recently, especially if we focus and think about GU and in kidney cancer, I think there's a lot of excitement around adenosine targeting. But just thinking in broad terms, when trying to target metabolism in this very complex tumor microenvironment, what are some things that you think about in trying to identify a good target, that where you are trying to perhaps either hit a pathway that is susceptible in the cancer cell and proves beneficial for the other immune cells, et cetera, that are in that microenvironment?
Yeah, it's a tough challenge. I mean, cancer metabolism as a field has come up with lots of targets, and then they run into problems because there's metabolic plasticity and cells can sort of switch around and change. And then the other thing that'll happen is a lot of times is you'll treat with a drug that will block a metabolic pathway and the cells will not die, but just maybe go into more of a stasis. So I think it's to be determined a little bit about ACSS2. It is interesting that, again, there's 3 or 4 ways you can make cytosolic acetyl-CoA, which, again, the product of ACSS2. And there does seem to be a metabolic vulnerability in the kidney cancer cells. Now, we'd really have to go into some different animal models and eventually into patients to see how robust is that going to be. But given the effects on HIF-2 and then the fact that we know HIF-2 is important from belzutifan and other genetic sort of analyses, this one might be a little bit different, right? Because it's not necessarily that you're at the top of a metabolic pathway that has many, many opportunities to sort of bypass. It's like this makes the product, and the product does things that have impact. So it's immediately adjacent to where the effector molecule is. So this one might be a little bit different. And if kidney cancer cells can't make this or don't efficiently make acetyl-CoA through these other possible pathways, then that would be a potential vulnerability. So we'll have to see how much the flexibility comes into bear. And there are drugs available for some of these other molecules, particularly ATP citrate lyase. So it could be a situation where maybe an ACSS2 plus an ATP citrate lyase inhibitor could have some impact. We know other cells can make acetyl-CoA through different means altogether, but those 2 things could have a synergistic effect. So I think the next steps are going to be to try to understand to what extent can the cancer cells evade this? What is their metabolic flexibility? And so far, Zach's data suggests it might be a little bit limited. And then what happens to other cells? And of course, the immune cells would be major direction there, but other cells in the tumor microenvironment, of course, as well.
Well, I want to congratulate you both. It's great work, and so thrilled that it's published on JCI and that we got to chat about it here today. Want to thank our audience for joining us here at GU Oncology Now, and hope everybody has a wonderful day.